how do we organize the staggering, just the spectacular diversity of life on Earth?
It's a huge question.
It is. I mean, the planet is buzzing with untold numbers of organisms, and if you as a curious learner want to make any sense of who's related to whom, you need a system, a logical one.
And that structure, that's the practical need for what we call biological classification or taxonomy.
Taxonomy.
Right. And it's much more than just, putting names on things. It's really a fundamental scientific endeavor that helps us reveal the history of life.
So it's like drawing a family tree.
Exactly. When we classify organisms correctly, we're essentially drawing that family tree, uncovering the true evolutionary relationships, and mapping descent from common ancestors.
Okay, let's unpack this then. Our sources for this deep dive focus on the massive evolution of that family tree. It's really a story of correction, isn't it?
It is.
Moving from these ancient, simple groupings to the system most of us recognize, the one from the late 20th century, Robert H. Whittaker's 5 Kingdoms.
Proposed in 1969.
Right. And Whittaker's system gave us the organization we learned in school, but it was revolutionary because it forced us to stop thinking about life as just plant or animal.
That 5-kingdom model was absolutely necessary.
Yeah.
The system before it was just structurally inadequate to handle the explosion of knowledge that was happening.
From the microscope, you mean?
The microscope, biochemistry, just understanding how cells actually worked, the old categories, they just crumbled.
If we zoom way, way back, you had early thinkers like Aristotle.
Right.
Trying to group life based on where it lived.
Yeah, land, water, or air. Very simple.
Then you get the organizing genius of the 18th century, Carolus Linnaeus. He gave us that traditional 2-kingdom system in 1758, Plantae and Animalia, plants and animals.
A great starting point for naming things, for sure.
But it failed almost immediately once we started looking closer than, the naked eye.
The limitations were massive. They centered on, I'd say, 3 of the most crucial biological characteristics. The Linnaean system simply couldn't differentiate between unicellular and multicellular organisms. And crucially, it failed to distinguish between prokaryotes and eukaryotes.
Which we now know is probably the single most important difference in cellular design.
The most important. And third, it lumped together organisms that made their own food, the autotrophs, with those that had to eat others, the heterotrophs.
So the green slime on a pond, a giant redwood tree, and a blue whale were all somehow sorted into just 2 buckets based on whether they moved or not.
Pretty much. And that's where Whittaker stepped in and said, hold on, we need more rules. He introduced 5 core criteria to classify life based on intrinsic biological differences.
Not just what they look like on the outside.
Exactly.
So of those 5 criteria, which one do you think was the biggest break from Linnaeus's thinking? Was it the mode of nutrition?
It absolutely was. That was his 3rd criterion, mode of nutrition. We now had, photoatrophy for plants, but we separated out absorptive heterotrophy for fungi and ingestive heterotrophy for animals.
And that idea that how you get your energy is more important than what shape you are, that was groundbreaking.
Completely.
Wait, let's quickly clarify that terminology for our listener. What does absorptive heterotrophy mean versus, say, ingestive heterotrophy or holozoic?
Oh, a great point. Holozoic or ingestive heterotrophy is what animals do. We eat food, we chew it up, we digest it internally.
Right.
Fungi, using absorptive heterotrophy, do the opposite. They release digestive enzymes outside their bodies onto decaying matter and then just absorb the broken down nutrients.
So it's external digestion.
It's external digestion, and it gives them a completely unique ecological role.
Fascinating. So Whittaker also considered cell structure complexity.
Prokaryotic versus eukaryotic.
Complexity of the body structure.
Unicellular versus multicellular, sometimes called thallus organization.
And then ecological lifestyle. So was it a producer, a consumer, or a decomposer?
Yes, and finally, phylogenetic relationships. The evolutionary history. He really sought to organize life in a way that moved from simple, ancient forms to complex, recently evolved ones. That combination created a much more natural, meaningful framework.
So Whittaker gave us the new rules. But every system needs a way to organize everything, like a filing cabinet. That's where the famous taxonomic hierarchy comes in.
The nested system, right? It's the structure that dictates how specific or general a relationship is. We move from the most general grouping kingdom down to phylum, class, order, family, genus, and finally to species.
And the key insight there is that every level you go down, the organisms in that group are more closely related than the level above it.
That's the core idea.
Which brings us to the 5 kingdoms themselves: Monera, Protista, Fungi, Plantae, and Animalia. These 5 groups captured life as we understood it back then.
And we can't forget the power of the binomial scientific name, the two-part name.
Genus plus species, which Linnaeus introduced.
Mm.
Like the common house cat, Felis domesticus. That name is precise. It's universally understood no matter what language you speak. And you can use it to cut through all the confusion of common names.
Oh, absolutely.
Like our sources note that a daylily, a mariposa tulip, and an Easter lily all share the word lily.
But they're not even in the same genus.
Right. But because scientific classification places them all within the lily family Liliaceae, we understand their broader, more distant kinship. It prevents scientific chaos.
And this system isn't just a record of what's alive today. It's also a vital bridge to the past.
How so?
Well, the classification structure includes extinct organisms. This gives us critical clues about evolutionary history. We can place dinosaurs within the scheme or look at something like the mastodon, a massive extinct mammal,
Great.
and see that it is classified as a close relative of the modern African elephant species, Loxodon africana. Taxonomy is history made manifest.
Okay, here's where the organization starts, with the deepest, most fundamental split in all of biology, the cell structure. Before we even talk about the 5 kingdoms, we have to talk about prokaryotes and eukaryotes.
That division is truly foundational. Think of it this way. Prokaryotes are these simple, efficient, stripped-down cells.
Okay.
They're defined by lacking a membrane-bound nucleus. Their genetic material, their DNA, it's just a naked, circular strand floating in the cell's central region. No internal rooms.
So the prokaryote is like a studio apartment.
Right.
Everything just happens in one large space.
That's a perfect analogy.
Yeah.
And eukaryotes, then, are the mansions. They're complex cells with a well-defined nucleus where the DNA is all neatly bundled up and protected.
And they have all those internal membrane-bound organelles.
Like mitochondria for energy or chloroplasts for photosynthesis, each one performing a specialized function.
So if the definition of a prokaryote is a simple cell without a nucleus, which of Whittaker's 5 kingdoms contained all of those organisms?
That would be Kingdom Monera. And I think the greatest conceptual achievement of the 5-kingdom system was successfully isolating Monera from everything else.
It was the first time all the simple ancient cell designs were grouped together.
Exactly.
So Monera is the exclusive club for all prokaryotes. Bacteria, mycoplasma, actinomycetes, and even cyanobacteria, which we used to call blue-green algae.
And they are incredibly diverse in their metabolism. Despite being so simple structurally, their cell wall is made of non-cellulosic material, usually peptidoglycan.
And their nutritional modes are all over the place.
All over the place. Some are photoautotrophic, living off light like plants. Others are chemoautotrophic, like nitrifying bacteria that use chemical compounds for energy. And, of course, many are heterotrophic, living as decomposers or parasites. Monera is where the ancient roots of all metabolism lie.
It's amazing that one kingdom contained all of that. Now let's flip the switch to complexity. How did Whittaker handle the other 4 kingdoms, the eukaryotes? Protista, Fungi, Plantae, and Animalia.
Right, so we've isolated the prokaryotes. Now, within the eukaryotes, we primarily differentiate the 4 groups based on 2 main things:
What are they?
the presence and composition of their cell wall, and again, their mode of nutrition.
Okay, let's start with the one everyone recognizes, Kingdom Plantae.
Plants are multicellular, eukaryotic, and have a rigid cell wall made of cellulose. And they are, of course, the primary autotrophs. They use photosynthesis to make their own food,
Simple enough. Then you have Kingdom Animalia.
which lacks a cell wall entirely. They're multicellular eukaryotes, usually highly motile, and they're always heterotrophic, obtaining food through ingestion, that holozoic method we talked about.
And then there's Fungi, the kingdom that really, disrupted that old plant-animal dichotomy.
Yes, and this is where the ecological insight really shines. Fungi are mostly multicellular eukaryotes, but their cell wall is made of chitin.
Chitin, that's the same tough material you find in an insect's exoskeleton, right? Not cellulose.
The very same. And more importantly, their nutrition is absorptive heterotrophy. They are the great decomposers.
So that separation wasn't just aesthetic, it fundamentally reorganized our whole understanding of food webs.
It did. It recognized that the ecological role of decomposition recycling nutrients back into the ecosystem is so pivotal and so biologically distinct that it deserved its own high-level group, just like production for plants and consumption for animals. That was a huge shift in the 1950s that Whittaker really formalized.
Which brings us to the last one, Kingdom Protista, the notorious catch-all kingdom.
The junk drawer of the system, exactly.
Yeah.
Protista includes all unicellular eukaryotes, so protozoans, phytoplankton, zooplankton. Whittaker was kind of forced to group them together.
But it created a really heterogeneous, a messy kingdom.
It was a major limitation. You had organisms with really significant dissimilarities, like a plant-like diatom and an animal like amoeba just sitting side by side.
They really do defy easy labeling. I mean, you have organisms in there that are purely photosynthetic, like diatoms. Then you have purely heterotrophic ones, like the amoeba,
And then the truly weird ones.
like euglenoids, which can switch their nutrition, they're photosynthetic when there's light, but they'll become heterotrophic scavengers if there's enough organic matter around.
That internal inconsistency was a huge clue that the map of life was still incomplete. And that's why classification is so dynamic.
It has to be.
The microscope helped change classification, but then came the invention of DNA analysis technologies. And that offered definitive, objective evidence to test these relationships.
This is where we shift from looking at cells and metabolism to looking at molecules, and where the map of life got a new, higher layer. In 1977, Carl Woese introduced the domain system, proposing a super-kingdom-level taxon based on molecular evidence, specifically ribosomal RNA analysis.
Ribosomal RNA, or rRNA, is critical. It's the universal molecular machine for making proteins in every living cell. The genes that code for it evolve very slowly, which means they act like a reliable blueprint for tracking deep evolutionary history.
So Woese used the sequence of the 16S rRNA gene to map the deepest possible divisions in life.
That's right.
And what he found completely reorganized that simple cellular kingdom we just discussed, Monera?
It did. The new system recognized 3 domains. Domain Eukarya is straightforward. It contains all 4 eukaryotic kingdoms we know: Protista, Fungi, Plantae, and Animalia.
But the real upheaval was splitting Monera.
It was. Kingdom Monera was split into domain bacteria, the so-called true bacteria, and domain archaea.
So Woese's molecular work proved that the single kingdom Monera was, well, obsolete.
Essentially. The archaea, often found in extreme environments like hot springs or high salt lakes, they're ancient prokaryotes. But the molecular evidence showed they are genetically and physiologically distinct from bacteria.
And their rRNA sequences suggested something truly shocking.
It was shocking to classical taxonomy. The data suggested archaea are actually more closely related to eukarya to us than they are to bacteria.
Wait a minute, that's a huge claim. For the listener, what does that actually mean?
It means that the old Kingdom Monera, which we thought was one unified group, actually contained 2 separate ancient lineages that had been diverging for billions of years.
And there's a term for that, isn't there? When a group isn't natural.
Yes. When a group fails to include all the descendants of a common ancestor, we call it paraphyletic. Monera was paraphyletic and scientifically invalid because it artificially grouped eukarya's closest relative, archaea, with an entirely separate lineage, bacteria. The new 3-domain classification finally fixed that error.
That is the crucial refinement. We moved from organizing life based on simple movement, thanks Linnaeus, to organizing it by complexity and nutrition with Whittaker, and finally to organizing it by deep genetic descent with Woese's 3 domains.
And that's the key takeaway. The value of classification lies not in its permanence, but in its continuous adjustment. It's a human system that has to adjust to the real, constantly evolving relationships between species.
But even after moving past the 5 kingdoms, we are left with a massive question that still challenges the entire map of life.
I think I know where you're going with this.
If classification is meant to organize living things, where do the non-cellular entities fall?
Ah, yes. The entities that Whittaker's system and the domain system both notably excluded: the viruses.
Exactly. Viruses and viroids, which are just infectious RNA particles without a protein coat, are nucleoprotein entities. They're often considered intermediate, something between living and non-living.
They can only reproduce inside a host cell.
Right. So since they defy the cellular organization criteria used across all the domains and kingdoms, they ultimately challenge the very definition of a living organism that our classification systems are designed to organize.
Which suggests the map of life still has boundaries left to define.
Perhaps right at the very edge of life itself.